Hello, my name is Lauren Bell, and I was a 2018 recipient of the North Pacific Research Board's Graduate Student Research Award. I'm honored to have the opportunity to share a little bit about how this award has shaped my academic career and propelled me to where I am today. A little bit about me to start. I'm a child of the Gulf of Alaska. I grew up in Homer, exploring the shores of Ketchimek Bay, as you can see here. I actually left Alaska to pursue my undergraduate degree in biology at Stanford University, but then returned for a master's program at the University of Alaska Fairbanks, where I worked with Dr. Katrin Eichen to study Arctic marine food web structure. I then moved to Sitka in Southeast Alaska to work as a research biologist at the Sitka Sound Science Center. My work there gave me the opportunity to start spending a lot of time underwater in these rich kelp forests around Sitka Sound, which you can see in this video. I started at that time to think a lot about how these important coastal ecosystems in our state's waters may shift in the future with climate change. But it was honestly difficult to find much research on that topic. And this was surprising to me, given how these highly productive marine ecosystems function as important nursery grounds and food resources for coastal food webs in our state. So I decided to return to school once more to study climate change impacts specifically in our high latitude kelp forests, this time under Dr. Christy Croker at the University of California, Santa Cruz. Now, in the first year of my PhD, as I was thinking about seaweed's role in Southeast Alaska, this image of kelp beds teeming with herring during the spring spawn in Sitka Sound kept coming back to me. As many of you are aware, herring occupy a crucial role at the center of the Gulf of Alaska's coastal food web. And herring, or their eggs and roe, have supported humans in this region since time immemorial. I really saw the need to understand how this habitat interaction between herring and seaweed might change in the future as a big need. So this is what I ended up focusing on for my graduate student research proposal. I ended up applying and receiving funding to investigate the effect of spawn substrate on Pacific herring roe development under future ocean acidification and warming. And I'd like to share just a little bit about that work and what it looked like. So as we're all aware, Alaska's oceans are expected to experience significant warming and acidification over the next century, with estimates of up to a 3 to 4 degrees Celsius increase in water temperatures and up to a 0.4 unit drop in pH by the end of the century. Unfortunately, there hasn't been much research that have considered the impacts of both warming and acidification on Pacific herring roe anywhere along their range. And further, there's really been no research on climate change impacts that has considered the habitat that these roe are found in. So herring eggs are laid and fertilized on seaweeds and seagrasses, and they stick to these substrates for the duration of their embryonic development until they hatch. And of course, these are living substrates. Seaweeds and seagrasses are removing carbon dioxide from the water as they photosynthesize. And so it's now been well documented how this process is raising the pH and oxygen levels within these beds that the herring roe are a part of relative to outside of them. So for this reason, I was curious whether these habitats could actually act as a refuge for herring roe under future stressful environmental conditions. So my project proposed to ask 2 questions. First, how will ocean warming and acidification affect some common seaweeds that are currently being used as herring spawn substrates? And secondly, how will the roe themselves be impacted by these conditions? And does association with seaweed alter any of these effects? So thanks to the funding from the Graduate Student Research Award, I was able to custom build a unique experimental system in Sitka that allowed me to precisely manipulate temperature and pH conditions to simulate these future ocean environments. I used this system for multiple experiments focused on the effects on kelps and on herring roe. Now, my kelp experiments ended up revealing species-specific responses to future environmental conditions. So for example, one of the major species that's used as a spawn substrate by herring roe is giant kelp, and it actually appears relatively resilient to future warming and acidification. But on the other hand, some dominant understory kelp species may end up being less abundant and less nutritionally dense for consumers in the future. So these impacts could not only disrupt what species are available as spawn substrates for herring, but also disrupt the food and energy supply that's available for other consumers that are also going to be facing metabolic stress in these systems in the future. The manuscript that's describing this work is currently in review and hopefully will be out soon. Now, for developing herring roe, I found that warmer and more acidic waters are going to be metabolically stressful to these fish. So developing herring were breathing harder under future ocean conditions, and they had poorer body condition on a molecular level when they emerged as larvae. However, interestingly, it's very possible that these environmental stressors could go unnoticed if researchers were looking at the wrong thing. So I found that higher temperatures and lower pH water alone had opposite effects on larval morphology. So this was indicating that both conditions cause stress in herring, but it manifests differently in how it affected their growth. So the result of this is that when herring are raised in both warmer and more acidic waters, which is the more realistic scenario in the future, the visual evidence of this stress effectively cancels out. So it conceals that anything is going on, even though we know that they are more metabolically stressed. So these results really point to the importance that future monitoring of this species integrates other metrics of health beyond just looking at their external morphology in the future. And it really has important implications for other fish species that we monitor in Alaskan waters moving forward. Now, sadly, my dream of testing whether seaweed habitats could act as a refuge for herring roe did not ultimately come to fruition in these set of experiments. The herring roe that I spawned onto kelp blades had surprisingly low fertilization rates and ended up sloughing off of these seaweeds in my experiment. Now, I was kind of scratching my head about this, given that this is the natural habitats you find the herring roe on in the field. But then I was talking to some subsistence harvesters of herring roe on kelp, and they pointed out that the herring roe aren't actually naturally found on smaller kelp blades in the wild. And I realized that in an effort to pick blades that were small enough to fit into my experimental trays, I inadvertently demonstrated that there was some kind of chemical interaction between fish roe and these really young seaweed blades, perhaps more chemically defended or something along those lines. So still a very valuable discovery. And I think that there is much more to consider about the interaction of this forage fish with its early life stage habitats in a changing ocean. I do want to point out that the research that was supported from these Graduate Student Research Award funds has had a pretty impressive reach. There will ultimately be 3 publications from this work. I've presented on this research twice at the Alaska Marine Science Symposium, as well as other national and international conferences. I've shared the implications of this work with tribal governments, municipal committees, and high school classes, and I'll be presenting some of this work this fall at the public festival, Sitka Whale Fest, if any of you are able to come join for that. This work was featured in an exhibit at a Sea Life Center down in Santa Cruz. And finally, there were some exceptional Mount Edgecombe High School students who volunteered during these experiments and have since gone off to pursue their own academic paths in science. So really, it is no exaggeration to say that the impact of the Graduate Student Research Award on my research and professional development has extended orders of magnitude beyond the specific dollar amount of this award. I truly credit NPRB for providing me critical support as I've built my research portfolio and my networks in this region that I call home. Now, I'm thrilled to share that after wrapping up my PhD, I did step into the position of research director at the Sitka Sound Science Center. I know that this means that my collaborations with the North Pacific Research Board are only just beginning, and I look forward to much more work with you all in the future. Thank you again for all of your support.